Okay, I'd like to welcome everyone to today's Wednesday seminar. Firstly, I'd like to acknowledge the traditional custodians of the lands on which I am speaking to you from today, the lands of the Wurundjeri people of the Kulin Nation, and also those are the lands on which many of you may be watching this seminar from. May I also pay my respect to their elders past and present and acknowledge their continuing custodianship of these lands for many, many years. It's my great pleasure to introduce Dr. Niall Geoghegan from my lab for today's Wednesday seminar. Niall's been working within my lab in the CDI since 2016. He joined us from John Coper's lab in the School of Engineering in the University of Glasgow, where he completed his PhD. And he came to us with a unique set of skills, having completed a master's degree in electronic engineering and then his PhD, in which he built a microscope platform to do turf phlegm and fluorescence correlation spectroscopy. And he studied the biophysical properties of cell membranes. Since arriving, Niall has worked closely with many staff and students within my lab and also the CDI, and he also contributes greatly to creating a very positive workplace culture. However, Niall is probably best known for his instrumental role in establishing our lattice light sheet microscopy program, and he's helped us to achieve something we never imagined we'd be able to do prior to 2016. And that is to build a custom built highly advanced microscope, which has allowed us to address important questions at least 5 years earlier than what we might have been able to do otherwise. So to highlight this powerful approach, he's going to speak about two projects today where he's played a significant role, including one where he's really helped us to form an understanding about membrane remodeling during malaria parasite invasion of red blood cells, work which has earned him awards at several conferences so far. So without further ado, please welcome Niall.
Hi everyone, thank you very much, Kelly, and thank you for a very kind introduction. It's an absolute privilege to be able to present this work to you all in this very prestigious forum. And I recognize the title of my talk contains a question. If a picture is worth 1,000 words, how much is a movie worth? And I think it's always really important, if you have a question in the title of your talk, to answer that question. I'll endeavor to do that over the course of this talk. But before that, I really need to tell you a story. And this story for me begins in 2014. So if I can just move that. So 2014, I was still a PhD student at the University of Glasgow. I was coming to the pointy end of my PhD, so things were starting to get pretty serious. And as Kelly mentioned, I was building various types of microscopes to look at different aspects of cell membrane biology in particular. One microscope was a turf microscope, and just really quickly, a turf microscope is a system which enables you to look at a very thin slice of a cell at the bottom, at the cover slip interface. As you shine a laser at an angle and it gets totally reflected, a small evanescent wave propagates into the sample, exciting fluorescent molecules within this close to 100 nanometer region at the bottom of the cell. So it's great for looking at the bottom of a cell, not great for looking at much else. But in particular, I was doing some work with some groups who were very interested in a specific endocytic pathway in insulin-regulated glucose metabolism. And they had this, and they were interested in looking at this in fat cells, but they had this HeLa cell ortholog, which they were keen to characterize to see if it was any use for further biology. So this is an example of what that looked like. And what I found was, as I started to image, more often than not, the cell would detach from the substrate and subsequently die. Now, at this point in my life, the word phototoxicity hadn't yet entered my vocabulary. And let's be honest, I was an engineer trying to do biology, and it was never going to start particularly well. At about the same time, I went to Edinburgh, and these two images are just there to show you just how beautiful Scotland can be at times when the sun's out. But I went to Edinburgh and I went to a one-day symposium on imaging the subcellular world. And this American guy got up and gave a talk, and he was called Eric Betzig. Now, some of you might be familiar with this guy. He came to WEHI in 2015 to give a talk, and he spoke about this revolutionary new technology in the live cell imaging world, and he called it the lattice light sheet. And he stood up there and he said, this is going to change the way that we do live biology. And when you promise something like that, you really think, well, the data has to stack up to the promise. And so he got up there and he presented the data, and it just kept coming and coming. And when I saw what he had to show, my jaw hit the floor. You know, it took me a while to pick it back up, from looking at the development of zebrafish embryos to the dynamics of the actin cytoskeleton to incredibly delicate processes like cell division and immune cell target interactions. And I sat there looking at this data and I thought, how on earth are they able to do this? You know, I'm killing cells with one thin sliver of light, and they're essentially doing what I'm doing, but across the entire cell and across multicellular organisms. So I went away and I looked at the paper, and it's easily one of my favorite papers, largely in part in how it starts. And one of my favorite quotes is, the hallmark of life is that it is animate. Every living thing is a complex thermodynamic pocket of reduced entropy through which matter and energy flow continuously. And I think this is just a great example of what happens if you let physicists write about biology. They can get a little bit carried away. But essentially, what the system was trying to get at was, in microscopy, we always played this trade-off game, where ideally, in the perfect world, we'd have a microscope where we could resolve single molecules in real time, whilst being minimally invasive to the biology, imaging deep into tissues with super high signal to noise. But this isn't the reality of the situation. The reality looks more like this, where you have to pick and choose certain elements of this trade-off. And this surface area of the shape is finite. So when you pull it in one direction, you're pulling it away from other parameters. So the way that we've been doing live cell biology for the last many decades is largely been using wide field and, importantly, confocal microscopy. Confocal microscopy is this wonderful technique where you're able to gain optical sectioning capabilities by only detecting the light interacting with the cell at your focal plane. So you shine a laser, it focuses it to this very small point, you scan that point across your cell, and you detect the light from that specific focal plane. You can stack this up in Z, and you can build three-dimensional models of cells. Now, it's a great way of doing three-dimensional sectioning, but it is a terrible way of trying to image live biology in real time, largely because the amount of light that you have to jam into this small, poor little cell is so high that more often than not, to get anything resembling a three-dimensional, four-dimensional data, the cell will begin to die before you can resolve your biology. So light sheet, and in particular lattice light sheet, is sort of this four-dimensional and fast imaging resolution. So the thinking behind light sheet is you take that sort of rugby ball or footy ball shape focus spot of light, you turn it on its side, you stretch it out in length and width, and instead of having a single point you scan very quickly, you have a sheet of light which illuminates your whole plane simultaneously. So it's much faster than point scanning confocal. The problem is, and this is a problem of physics, when you stretch out a Gaussian light or a focused light spot, it's a Gaussian intensity distribution, the longer it gets, the thicker it gets. So you lose resolution in Z. For subcellular dynamics, this becomes problematic. So Betzig and his team, as physicists like to do, started mucking around with different types of laser beams. So they thought, well, how about a Bessel function? So a Bessel function, you have this tightly constricted, thin central region of high intensity laser light, and then these circulating and radial side lobes coming off the side. And so in context of stretching out your Gaussian light sheet, you can get a very thin Gaussian light sheet, but it'd just be quite short. If you stretch that out longer, it gets a bit thicker, but with the Bessel beam, you can get the best of both worlds. You can get a long and thin, in essence, high resolution light sheet. The problem is these side lobes. So these side lobes still are light and interact and will give you out of focus light. So what Betzig and his team, again, as physicists do, is they did some simulations and brought some Bessel beams closer and closer together. And as these Bessel beams constructively and deconstructively interfere with one another, they eventually reach a point of tight constriction, which we call a lattice light sheet. And the way we actually build up an image with this lattice is by parking it in the sample itself. And instead of moving the light, we move the sample, and we move it very, very fast. So you're able to build up information of the subcellular world with very, very high spatial temporal resolution. So now that I understood how this worked, I thought, wow, gee, it would be great maybe one day to be able to get involved in this technique or maybe contribute it in some way. And I think in storytelling terms, that's called foreshadowing. But in 2015, on the other side of the world, here at the wonderful Walter and Eliza Hall Institute, Kelly Rogers and Lachlan Whitehead, otherwise known at the time as the Imaging Facility, they were having a very similar epiphany to myself, and that they recognized that this technology was just absolutely going to shake the game up in terms of live cell imaging. So they thought, how can we get our hands on it, bring it to the Institute, and then bring it to those who are going to get the most out of it, i.e. the biologists. So at the time, there were a few ways that you could get your hands on it. One, you could take your research to Genelia Research Campus, where the system was invented, and you can run your experiments there. And that's a great experience. It's like a Disneyland for microscopists. So I still haven't been. Kelly and Lockie have been many times, and I'm not bitter about that. Oh, I am a bit. The other option was there was a pre-commercial version at the time from a company called 3i. And this was not really the best option at the time, because it was quite cost prohibitive. It was, about twice the cost of the parts that built it. At the time, 3i had zero support locally in Australia, so it would have been a risky purchase, and you would still need to employ someone to come and sit on it and babysit it and run it. The other option is you could build it yourself. Now, this was made quite easy because Betzig and his team recognized that there's going to be this gap. They're going to invent a tool, and then they're going to license it to Zeiss, and Zeiss will spend anywhere from 5 to 10 years turning it into a turnkey solution. But in that time, you need it to be in the hands of those that are going to do the most with it, the biologists. So they had this program where you could send someone over for 2 weeks, and they would sit you in a room, in a classroom, with other people to learn how to build the system, screw by screw, lens by lens. And so Lachlan went over to Genelia to do this. And when he came back, Kelly said to him, "Do you think we can do it?" And Lockie's exact response, and I quote, "I don't see why we can't." Which is such a wonderful response you'd expect from someone with a double degree in physics and political sciences. But because he said that, and I'm very pleased that he did, because it meant that I got to move to this beautiful country and we got to build it ourselves. So this is what that looks like. Obviously, we worked this quickly. There are some cameo appearances in the middle of this. I think Alan Cowman joins us halfway through and actually screws in a part. I do remember my first few weeks here, every time I passed Alan in the hallways, he wouldn't say hello, he would just say, "Is it built yet?" And so that's at the point I recognized the pressures that came with working at WEHI. So come 2017, so now we have the system, it's working. What are we going to do with it? And at the time there was this project that was a sort of mature point, and this was looking at cell death, and in particular, back-to-back mediated intrinsic apoptosis. And it was known widely that during this process, that mitochondrial DNA can, under certain circumstances, be released and trigger a signaling cascade and some inflammatory downstream effects. Also, mitochondrial DNA itself has been found outside the mitochondria in various infections, infectious diseases. But the exact mechanism by which this process could occur was pretty poorly understood, and it had never actually been captured in real time. So this was work undertaken largely by Kate MacArthur in the Kyle lab at the time, and side by side with Lockie and Kelly, and eventually myself as well. And so the idea was, can we actually apply these imaging technologies and capture this this unknown event, this elusive event? So in the early days, Kelly and Kate tried to work this up on what we had available to us at the time, which was a resonance scanning confocal system. So here you see in red the mitochondria, and the green is the mitochondrial DNA. And the one thing you'll notice is it's very noisy, and that's basically because in order to try and preserve the sample's viability, but also get the temporal resolution that you needed to image the event, you had to make sacrifices elsewhere. So you sacrifice the signal to noise, and as a result, you actually lose resolution. But with a lattice, you make less sacrifices, and when you make less sacrifices, your science is better. So here we can see the mitochondrial DNA in green, and also the mitochondrial membrane labeled here in red. And so this looks more like what the system should look like. And just by virtue of making fewer sacrifices, you can start to answer these questions. So when you take a cell and you induce apoptosis through a drug, it goes through a few very well controlled steps. So first, the mitochondrial network will start to break down, and the sort of long sausage-like mitochondria will fragment and become round. At a certain point downstream of this, the mitochondrial DNA was actually seen to just pop out of each of these individual little spheroids. This was remarkable. You know, no one had ever seen this before in this level of detail, or even at all. And what was striking was, A, the mitochondria came out, but it stayed quite proximal to the mitochondria itself, and B, it seemed to only escape through a single point on the mitochondria. It didn't just come out and become diffuse. And so through previous work that Kate and Ben's lab, they knew that this was a back-to-back mediated process. So then they decided, well, then what do we do? Well, we label backs and back, or in this case, in this video, backs. Watch what happens. So it starts off as a cytosolic protein, which as the cell dies, it becomes punctate and locates to the mitochondria. Now, if you zoom in on this and you have a closer look in 3D, you see that both back and backs locate specifically to the constricted region with the DNA squeezed out. Because lattice gives you this beautifully broad overview of a complex process, it is limited in resolution, but what you can do is you can figure out from the lattice exactly the specific point where you think the interesting biology is happening, and then move to fixed imaging and look at it using super resolution microscopy. So here we have some structured illumination microscopic images from our SIM platform, and they found that these pores were these huge macropores through which the mitochondrial DNA could escape. Using this technique, then they could place this in the broader context of the cell death process itself and look how it related temporally to things like cytochrome C release. And they were able to find that these temporal kinetics were really well conserved, and they occurred at very specific time points. Now, when I came to this project, the work was quite mature, and it was at a point when Kate had submitted all of this, a majority of this to Science, and Science had come back to her, as Science does, and said, wow, this is fantastic work, but could you also do X, Y, Z, all this other 100 terabytes worth of experiments? And one such experiment was to look at how these processes related to the breakdown of the mitochondrial membrane potential. So we could do that because there's some fluorescent dyes that you can apply where they'll basically fluoresce in a region of increased membrane potential, and the mitochondria has very high membrane potential. So here in cyan, we have our membrane potential sensitive dye, and it did disappear, but it was a lot more interesting than just being there or not. You actually see it begins to blink at a single mitochondrial level. It sort of flashes on and off as the cell begins to die. Now this, I don't know if this is particularly relevant. This is just an example of the blessing and the curse of this type of technology. Is you can start looking at one thing and you can start to see several others, and it can take you off in some really strange tangents, but it can be quite exciting. Also here, you see a cell in the bottom right corner, the potential disappears, but also downstream of this, just before the cell fragments, it comes back on in a punctate way and then disappears. Again, not sure what this means, but it's the kind of thing that, I thought it would be nice to throw out there, because someone out there, and especially this particular audience, might say, oh, wow, that's this, or that might just be nothing. But to summarize this point, the technology really allowed Kate and Lockie and Kelly and Ben to visualize this process in exquisite detail, and it had never been before done. And also, because we had rapidly adopted this technology, the point when Science turned around and said, can you do all these extra experiments, rather than having to get back on a plane and go to Genelia, you could just come to level 4. And Kate turned to me and said, oh, I need to basically run this system into the ground for 3 weeks. And I was like, fine, let's do it. So at this point, it was really good for us, because now we knew the system worked, and not just worked, but it stacked up against those that invented the platform, because we could put our data side by side, and it was, well, a bit biased, but I think ours was a bit better. So the question was, what should we do next? And that takes us to about 2018. At this point in time, I'm going to switch gears completely and talk about malaria. Now, I've never felt imposter syndrome as much as I do right now, presenting the life cycle of the malaria parasite to everyone here at WEHI, but here we go anyway. So it's a very complex life cycle occupying two different hosts, the human and the mosquito. And an area of really intense research, specifically, particularly here at WEHI and in the broader field, is the asexual blood stage. Now, this is primarily because this is at the point in the disease when someone would present with symptoms. So it's a very critical target for therapeutic and vaccine development. So this process occurs over about 48 hours. You have the invasion of a single red blood cell with a single parasite, and over the space of a couple of days, that multiplies into anywhere between 20 and 30 daughter merozoites to repeat that process over again. And in particular, the process of merozoite invasion of red blood cells has been really widely studied, because if you can stop that entry into the door, then you can block transmission of the disease. So it's a really exciting point, and it's something that Kelly, in collaboration with Alan's team, have been trying to look at with varying microscopy techniques for the last decade or so. So the invasion process itself, a lot is actually known about this process in a lot of detail. It begins with a weak attachment to the erythrocyte surface of a single merozoite. The merozoite then deforms the membrane and reorients until its apical end is orthogonal to the plane of the membrane. At this point, it forms a fusion pore via the RH5 bathogen interaction, and then through which it can form the tight junction. This is sort of the nexus through which it invades, eventually pinching off behind it its new home, the Parasitophorus vacuole, and inducing acanthocytosis. Now, this is a really challenging thing to study for a few reasons. One, the parasite, it's very small. It's a single cellular organism, which is a micron in size, but with subcellular organelles. So you can imagine in terms of light microscopy, that can be a bit of a challenge. The process is quick. This is done and dusted in about 30 to 60 seconds or so. And it involves numerous ligand receptor interactions. So it's a very molecularly complex process, and also biophysical changes in the host erythrocyte. So ideally, you'd want a technique in which you could study this in 3 dimensions in real time, but with the molecular specificity afforded to you through fluorescent labeling, hence light microscopy. So the sort of standard in the field until now was just traditional transmitted light DIC microscopy. So here we have a movie, and you see a parasite interact and deform the membrane here, and eventually reorients itself until it nestles itself nicely into the erythrocyte. And just through this really simple imaging technique, we've learned so much about the process. But this comes with its challenge, mainly due to its inherent two-dimensional nature. And the red blood cells themselves, they're they're wonderfully 3D, and they have a very well-regulated shape, so you know what they should look like. And you can't guarantee the parasite is always going to invade the right way within the plane of your imaging. So in 2017, Cindy Evelyn started as an honors student with Kelly, and her project was to image this process in 4 dimensions on the lattice light sheet microscope. Unfortunately for Cindy, due to some Scottish engineering incompetence, the system wasn't quite ready for the start of her honours year, but she did manage to work up quite the protocol on our resonance scanning confocal microscope. And this is what she managed to do. She managed to get more out of this technique than really should have been possible. And you can see here as we go side on to the cells, even though we've got some 3-dimensional information, so many sacrifices had to be made in that dimension just to go fast enough that the cells, they look more like cinnamon rolls, when as we know, they should look like donuts. So when you transfer that to the lattice, because eventually Cindy was able to do this, this is what it looks like. We have mature schizon here in cyan and the erythrocytes in magenta, and eventually we see egress of the mature schizon. The merozoites fly out at varying speeds. We have a deformation of the host cell. We can cut away the top. We can look at the internalization of the parasite and the subsequent sealing behind it of the vacuole as it sets up its new home. and then acanthocytosis. We can use different combinations of dyes on a dual camera system and go a bit faster. This is easily my favorite data set that we've ever taken. And you see this almost like it's bouncing off a soft pillow as it reorients itself and internalizes, and then the ensuing acanthocytosis. So this is fantastic. We had this amazing technique which we can then probe this complex process in 4 dimensions, and so we can start to ask some questions. Now, one of the questions of the field which we thought we would tackle first was the origins of the Parasitophorus vacuole membrane. Now, the reason this is the question is because the parasite brings with it some lipid material and membrane proteins. And there's been questions surrounding whether the PVM is completely made of host material or if it's completely made from parasite-derived material or a combination of both. Now, this controversy seemed to stem, as far as my research can say, mostly from the early 90s, when you had a few papers who were like, it's completely host, and then on the other side, you had a few papers saying it's completely parasite. But on the parasite side, it was really just one group, so I think they really have thrown a spanner in the works. But for us, we thought we can measure this because it was quite clear from our data that it's pushing in the fluorescently labeled membrane, which we'd only labeled the erythrocyte membrane. So we thought, how about we look at the surface area, because we can, of the infected erythrocyte, and does that change with enough? Does that change by enough to compensate by the formation of PVM? So we generate this surface mesh, and now we have basically transformed our morphological phenotype into a spreadsheet of numbers. And as a result, we can look at how that surface area tracks through time. So here you have an example where a parasite invades from the left, and it comes in, and you've got the surface area on the right, and as that comes in, pinches off, that graph drops by a stepwise shift in microns squared. And we thought, wow, that's a wonderfully clear shift. Sometimes a cell can be invaded by more than one parasite. So here we have one parasite invading, we've got one stepwise change, and then the next parasite comes in and we get a second stepwise change. So we thought, yeah, this is pretty clear. It's pinching off quite a bit of the host cell. And when we look, if we add that back to what's left of the host cell, almost always it came quite close to just being the amount of surface area that was there previously. So we thought this is pretty striking. Another thing that we then looked at was the role that calcium plays in invasion, because it's been widely reported there's this small punctate calcium flux that happens if you load the erythrocyte with a calcium sensitive dye, and at the point of RH5 basogen interaction, it forms a pore, and then the calcium dye can flush back into the parasite, and you get this bright flux. So here you'll see that in this movie here, as the parasite deforms the membrane as it comes in from the right-hand side, we get this bright yellow flux, and then the parasite internalizes. And what Phoebe found, who was an honor student at the time, was that this flux always coincided with the final deformation of the membrane. So the flux, so any deformations that came after the flux, that was an attempt at invasion, or it was an invasion itself. And it always occurred on the parasite side, so that we knew the flux was happening in the parasite, we knew when it was happening, and it also gave us now a temporal marker by which we could then assess the effect of downstream inhibitors of the invasion process. So this was a pretty useful tool for us to then use, and that's what we did. So one such inhibitor is R1 peptide, and this inhibits the AMA1 RUN2 interaction, which is at the point of formation of the tight junction. So in the sort of diagram at the bottom here, you see we have the recoil, final recoil, followed by the calcium flux. And eventually, you see this big attempt at internalization in the acanthocytosis. So here we can see that in this movie here, the parasite comes in from the side, it pinches back and the flux is in green, and then immediately following that, it drives itself in, buckling the cell under the force of that movement, and then acanthocytosis happens, which was pretty interesting. And what that suggested to us was that even in the absence of the tight junction formation, it was still driving itself in as it thought it was trying to invade. So what we can do then next is we have the surface information, and we can look at the Gaussian curvature of that surface. And this is a really useful tool at looking at the nature of that saddle-shaped curvature that occurs at the rear of the parasite as it invades. So under normal conditions, as it invades, you get this really steep, a really high degree of negative Gaussian curvature at the point of invasion. When we inhibit with our own peptide, following calcium flux, as it drives itself in, you don't see that degree of deformation. So it is trying to invade, but it just can't deform the membrane enough. So it can still attempt it in the absence of a functioning tight junction, but also this is suggesting some implications for this interaction and in disrupting the underlying cytoskeleton. Because the parasite is still exerting its motor force, so that force should be relatively conserved across the parasites that we look at, but it can't deform the membrane enough, and really to do that you need to disrupt that anchoring of the cytoskeleton to the membrane to form the PVM. So then we had a look closer at the activation of the actomyosin motor of parasite, and we inhibited this then with cytochalasin D. And so when we did this, we saw the calcium flux because we still had that upstream pore-forming interaction, and then not much happened. So it can't deform the membrane, it can't attempt to invade, but it does still induce a canocytosis. So you see that in this movie here, where we have the rupture of a schizont. Parasite attaches, fluxes, it just sits there for a while doing something, and then a canocytosis. So the fact that a canocytosis was still happening in the absence of invasion, we thought was kind of interesting. And what causes and drives a canocytosis is actually not quite well understood. And so when we inhibit here, we can still get it. And when we inhibit here, we can still get it. But it is known that if you inhibit this interaction upstream, RH5 and bathogen, you can't induce a canocytosis. And we have this thinking that this is largely because at this point, when the pore is formed, the parasite is able to release the contents of its rock-free organelles, these little club-shaped organelles, into the cell itself. And so we wondered, is it maybe that the contents of these rock-frees are contributing to the process? So there was this paper back in 1978, this seminal work by Aikawa with this absolutely stunning EM microscopy where you can see the parasite captured halfway through the process. And highlighted here, they have the tight junction at the sides. In the front, they've got this little apical sort of attachment of the erythrocyte to the parasite, which they called the common duct, which I presume is probably this pore that we formed. So it's a physical connection between the erythrocyte membrane and the parasite. And in 2002, and the great thing about red blood cells, and easily the best cell out there, and my favorite by far, is that they're really well characterized because they're quite simple in terms of their molecular makeup and their composition. There was a group who are a bunch of computational biologists who were looking at these shape transitions in erythrocytes, and they found that just tweaking a very simple parameter of the ratio of the surface area of the outer and inner leaflets of the erythrocyte membrane could account for all these common shape transitions, including in a echinocyte. So we want to see, is there any evidence of any material from the rock freeze being inserted directly into the membrane itself? And so at the same time, we got this lovely parasite line from Danu and Tony Triglia and Alan's group, where they had labeled RON3, which was a rock-free protein with neon green, and RON3 basically stands for rock-free neck protein, but it's actually misnamed because it locates to the rock-free bulb. And using structured illumination microscopy, we could show this, and we actually saw that not only was it in the rock-free bulb, but it was in the periphery of the rock-free bulb, right, the rock-free bulb membrane. So we thought, well, this could be a useful tool for looking at the transfer of membrane-associated lipid from one organelle to the host cell. And so when we look at invasion, in normal conditions, the parasite comes in, and that punctate signal ends up becoming quite well labeled around the vacuole of the parasite. And the magenta here is RON3. And so in this case, you've got two images side by side, one with the membrane on, one not showing the membrane. And this is with armor peptides. So the parasite can't enter, but it can still form that pore. And as it tries to invade, it induces a canocytosis. And as you see it continuing to interact with the host cell, you see this RON3 signal start to localize to the erythrocyte membrane. In addition to this, we saw these incredibly striking structures when we inhibited the process, these membrane structures. So when we inhibited with R1 peptide, we saw these huge membrane tethers pulling back off the erythrocyte. And with the erythrocyte being the only thing we've labeled here, we think that this is as a result of a release of some lipid content from the parasite to the host cell, buckling, inducing echinocytosis, but also forming these large tethers. When we inhibit with cytochalasin D, see a similar structure, but it's now on the cellular side of the membrane. And this is because when we inhibit it, at this point, the tight junction is still formed. So it's able to form irreversible attachment. And as we zoom in, we see it's formed this quite large protrusion to the cell as it's depositing its content into that membrane. And so we thought maybe there will be some differences in the kinetics of the echinocytosis process under these different conditions. And because we had the calcium flux as this reference marker, we could say from this point, when the pores formed, how long until the onset of echinocytosis. So if we just disrupted the membrane itself by depleting membrane cholesterol with a methyl beta cyclodextrin, we saw that the echinocytosis process was increased. Under conditions of R1 peptide, there was actually no difference, which was quite interesting. And then when we inhibit with cytochalasin D, the echinocytosis process takes a lot longer. And this could be because that additional material is actually being pushed through past the tight junction. And it's known that the tight junction acts as a sort of filter and stops membrane-associated proteins going back into the erythrocyte, but not lipids. So lipid content might be going, just not the proteins, hence why it takes longer. So to summarize this section of the talk, we've now shown this wonderful approach of using lattice light sheet microscopy to image this really delicate and quite quick and complex process. We're able to quantify this and answer long-standing questions around the formation of the Parasitophorus vacuole, and we're able to come up with new ways of measuring and trying to tease apart some pretty interesting ideas about biophysics and the biomechanics of this process. So this brings us up to 2020, and yeah, it's been a year. But one of the things that happened quite early in 2020, before everything went a bit downhill for everyone, was the lattice light sheet came of age. So when Betzig and his team initially invented this system, they realized that they could commercialize it because it is a game-changing technology. So they licensed it to Zeiss, and in the meantime, they tried to generate Zeiss's user base by getting everyone hyped up, jumping over the valley of death in the technological hype cycle. And so what Zeiss eventually came up with was they turned this absolutely magnificent machine into this microscope. It's definitely a microscope. It does resemble a sandwich press or a photocopier. And the first thing you notice is the first thing that I noticed is that it's quite visually unremarkable. But what Zeiss have managed to do under that unassuming cover is quite impressive. So the biggest barrier to entry for the lattice has been the complexity in which interacting with the system has been. And, you know, it's not a conventional sample form. And so what Zeiss have done is they've turned the whole instrument upside down. And now you can approach it as if it was a confocal or a wide field. You can take any sample in a 96-well plate, 8-well chamber slide, dish, whatever, your standard format, just whack it on, press the button, go and come back. And this is fantastic. It's an absolute game changer. And so what this means, and I'd like to sort of like show you an example of where that really comes into its own. And so in order to do so, I really need to highlight one of the biggest limitations of the system we built, and that is where the sample sits. So you have these two lenses here, which cost more than I would like to admit, and you want to avoid damaging them. But your sample sits on this tiny little blue piece of glass in here. And that cover slip is about 5 millimeters in size, costs about $1, which I think is a bit expensive for the amount of glass you get. And then you have to image in that tightly confined space. This also sits in a bath filled with media, just filled with about 8 milliliters of media. So you can imagine if you're using an expensive drug or reagent, it's a bit cost prohibitive. And in the context of the malaria experiments, we're also limited by our field of view. So here's an example of an experiment that Cindy was doing where she was looking for invasions. And because you don't know if a particular Schizon's going to rupture, you sit and watch it for a while. If something happens, you keep recording. If nothing, you got to stick or twist and move. And she moved just at the right time. She caught this egress, and then she moved again, and she caught this invasion event where you see 2 invasions there. And I'm so glad she did, because this is absolutely from my favorite data set, but she almost missed it. And you want to, and basically you don't want to have to rely on luck in order to get your results. So now this is our field of view on the lattice that Zeiss have put forward. We can see this massive carpet of cells and chizons, and we just press go. We see egress happening, we see invasions, we walk away, we come back, an hour later, we harvest our data. If we play that back, you can actually see where the invasions occur. There's one there, there's one there, there's one there, there's another there, and there's another there. Now that's 5 in just one 20-minute run. And if you were to get 5 in a day on the other system, you would have heard dancing and singing coming from the room. So that's 2020. Now what about 2021 and the future? Now, because the Zeiss system has really basically filled the void and brought something to the table, which is going to absorb probably about 90% of the existing applications on our home-built system, what do we do with the home-built platform? And this is the beauty of having customizable tech, because here you've got a microscope. It's not just a black box that becomes redundant as soon as the next toy comes on the market. You've got a bank of lasers, which are really, really great. You've got some really expensive camera technology, some really high quality optics. So we can take this and we can do the next level of development with it. And just to give you some examples of that and where this technology has been taken by various groups is to, firstly, we could take 7 lasers and then try and do hyperspectral imaging of 7 interacting organelles all at one time. Or we can, oh, my PowerPoint just crashed there. That's not great. Hang on a sec. All right. Almost there. Amazing. So yeah, as I said, we can try and do these incredible things as we take it to the next level. On the left there, you've got 7 interacting organelles all together. I don't think I'm sharing my screen actually. I'm just going to bring that in. Right. But yeah, the question of where we take this next is really a matter for the users here at WEHI. We could look at single molecule methods where we can start to tease apart the ultrastructure and nanometric precision across the entire cell, as Wesley Gant did in his Nature Methods paper in 2016. On the right there, we can take it to, we can add some adaptive optics and start to image these subcellular dynamics in living organisms like zebrafish. But just to finish up here, this is, sort of to try and mirror that slide where I showed where Eric Betzig got up and spoke about all this incredible applications where they'd taken it to various groups and done all these things. And here at WEHI, we're so well placed to do that as well. And we are doing that, and across various fields from cell migration to cell death, to cell-cell interactions, cell division, immune cell, target cell interactions. Really is, the best place to try and situate these technologies. And yeah, we're in a really, really lucky position where we can do that. And I absolutely attest to the fact that I'm pretty sure I have the best job here, where I get to work side by side with all of you and try and visualize these incredible dynamic processes. So I'd just like to finish up by acknowledging specifically Kelly and our team. Kelly for taking a bit of a punt on bringing me in when I was coming from an engineering background, and I'm really glad that she brought me in so I could help out in this project, and I got to fulfill my dream of working on the lattice light sheet, which is pretty good. Cindy really has been a huge driving force behind the invasion work, and her patience and expertise in this area is second to none, and I don't think anyone else could really do what she does. She came to us as a physicist at undergrad, and she's this incredibly accomplished microscopist and parasite biologist now as well. Mike, Lockie, Verena, the other members of the team, and Phoebe as well, who did a lot on this work, and Sabrina, Nina, David, and Sean. And everyone at the Cowman Lab, who have really guided us with their expertise, specifically in the malaria work, and all these groups at the end here who I had some data on that final slide. Now I'd just like to finish because I did say I'd answer the question. How much is a movie worth? Right, so if a picture is worth 1,000 words, how much is a movie worth? So let's think about it. So we'll have a 10-minute movie, like we would have for an invasion movie. We'll do 2 channels, 2 colors, with an imaging volume of about 6 seconds per imaging volume. So basically you end up with about 100 volumes, 2 channels with 834 individual images times 1,000 words, which is about 168 million words, which in the SI units of word count, i.e. copies of War and Peace, that comes to about 278 copies of War and Peace, which looks a little bit like this. And we can thank Lachlan Whitehead for the animation. His talents as an image analyst are absolutely wasted. He should do this professionally. And I'd like to thank everyone for listening, and I look forward to taking any questions.
Thank you very much, Niall. Wonderful talk and great overview of lattice light sheet microscopy and the red blood cell project. I'd like everyone to give Niall a virtual applause if you can. Thanks, Niall. Great talk. So we have some questions in the chat here, and I'll start with a question from Alan Kalman. Great talk, Niall, especially the second half. Can you label the red blood cell cytoskeleton during invasion to see the local disruption as the parasite interacts and moves into the red blood cell?
Yep, I think we can do that. There are some nice live cell labels for looking specifically at actin, which we're hoping to do soon. That's really the only element of the cytoskeleton that has a suitable label for this, but it is one of the known components which is meant to be heavily disrupted. So that's the start.
Okay, thank you. Anonymous, how long does it take to do one full Z stack, for example, 1 millimeter cubed organoid tissue sample? If you were imaging something larger and constantly moving like a developing embryo, would the scanning happen too slowly compared to the motion of the cells?
So in the context of the lattice, the largest volume we can do is about 100 microns in length. So maybe 100 by 50 by 100, or at least on the new system, we can do 250 by 250 by 100. We are limited in depth in the lattice. It's really geared towards single cell biology. But if you wanted to go to the sort of larger tissue, we actually have a XYZ1 light sheet, which is specifically geared towards that. And they image so quickly that motion artifacts are really not an issue. Each imaging plane can be acquired in milliseconds. And so you can get across a large volume in seconds. And yeah, I think on that system, there's some incredible data of developing embryos.
Thanks, Niall. So a question from Harry Dempsey. Hi, Niall. I was wondering, what do you think about the role of machine learning in analysing these lattice light sheet microscopy videos?
It's a great question. So there's a few places where machine learning can really assist us here. Machine learning has its place in, I suppose there are a few places it can be pointed. So there's processing, analysis, and classification, as you will. The latter ones are a bit more of a challenge, but we've definitely had some real joy in applying machine learning for denoising, for example. Lockie and Nina in the imaging analysts have done some great work there. But also it can be used in trying to simplify these experiments, perhaps, in terms of having smart feedback microscopy where you can use machine learning as a classification tool to, say, find a Schizon that's about to egress, for example, so you don't have to search around like Cindy was doing frantically for the last 3 years. So yeah, it's got its place, absolutely, but it's yeah. There's a very blank canvas there if you want to get involved.
Okay, a question from Chris Tonkin. Great talk, Niall. On one hand, you show that most of the Parasitophorus vacuole membrane likely comes from the erythrocyte, yet you argue that lipids are coming from the rock tree to change the membrane curvature.
Yes.
So which one is it?
So Chris was reviewer 2 then, was he? That must be. Yeah. No, Chris, that's a wonderfully astute observation. I think what we're saying with our surface area measurements is that the PVM's ultrastructure is predominantly made from pinching off the host cell membrane. Now, within the resolution of our technique, we can't get down to, say, contributions of a certainly small molecular fraction. So we can't argue against some contribution from parasite lipids, but they may serve they may be more served downstream of invasion, because sometimes in occasions when we see it invade, it begins remodeling instantly. And you sometimes have occasions where the vacuole forms and you get a huge long protrusions as this sort of amoeboid stage starts to form. So I think the lipids definitely serve functional roles. They help in stabilizing the curvature or in the sealing process, or to undo the echinocytosis process. That's something we're thinking about exploring as well. But yeah, it's still a bit muddy.
Okay, another question from an anonymous person. Awesome. I wondered whether it can be used to visualize the binding of antibody to antigen. If yes, does it use the same marker like in the immunostaining?
I think, yeah, we could definitely try that. We've tried some experiments where we've had fluorescently labeled drugs dropped in and they bind to certain receptors and cluster. And because of the sectioning capabilities, you do get a really good signal to the background. And as they stick to the cell, you get bright spots. Ideally, you'd have fluorogenic probes that only fluoresce once they bind, but they're a bit harder to design, less common. But I don't see why not. If it's a surface receptor, probably.
A question from Justin. Niall, that was beautiful. Does your data preclude the notion that merozoite lipids contribute to the PVM? You showed tethers and mentioned they may come from merozoites, but what about the PVM? Yeah,
I think that just goes kind of back to Chris's question as well about the relative roles of these two membrane contributions. I think the parasite, if it can, will just use as much of the host as possible to form that PVM. And we have some data that shows quite early after invasion that the lipid composition of the PVM is different to the erythrocyte. And actually, once it gets to the ring stage, in other papers, they've shown the surface area comes back up to where it was before. So that lipid may be used in trying to undo the damage it's done after it invades, but something that we would love to explore, and we could image a bit longer and try and figure that out.
Another anonymous says, wonderful talk. Another anonymous says, great seminar, thanks. Can you do high throughput imaging, 96 or even 384 well plates with the new lattice light sheet microscope?
We will be able to when Zeiss finally released the software update that allows us to do that. We have the plate format, we can put it in, we just can't do any automation yet. Problem with that is it will be 96 times 168 million words. So, your copies of War and Peace will get a lot bigger, so you need a lot of hard drives. But yeah, absolutely. And we're actually starting to look at, is there space for a 4-dimensional type of screening?
Anonymous, Ashley says, beautiful talk. That is all. Another anonymous says, great talk, very accessible to non-experts. Thanks. And finally, last question is from Tony Burgess. Is the computer hardware limiting for handling these data sets?
That's a very loaded question, because I think Tony knows the answer to that is yes. There is the biggest bottleneck we are facing by far is the data, not just analyzing it, but processing it, because it comes at a weird angle. We've got to deskew it, deconvolve it, and we've got to open it in something. We've got to visualize it. When things are 3D and 4D, visualizing them is actually a bit of a challenge. Use some really intense graphics processing, but we have the hardware place to do this. But there are some bottlenecks in the pipelines, which means, traditionally you walk up to a microscope, you take your images, you know if your experiment has failed or gone well that same day. But with a lattice, it could take maybe a day or two afterwards. We're working on that. We're working on that.
Well, that's it for the questions. Thank you very much, Niall. I'm sure everyone agrees with me when I say that it was a fantastic talk, really lovely seminar, and another virtual applause for you from everybody. And thanks again. And thanks everybody.
Thanks everyone. Bye-bye.
Bye.